Hi everyone. So first of all, I'm so sorry that I'm not going to be able to be with you guys for Wednesday's small groups, but I'm hoping that this also is very helpful. And remember that with the email that you received or looking at announcements, there's also a link that's going to give you your own copy of this PowerPoint presentation so that you can follow along with the video. So I'm going to walk you through the video and then I will let you know when you should be heading over to your own copy to be trying out the slides and manipulatives and to check your understanding. All right, so let's go ahead and get started. So last week we talked about mitosis and meiosis as a whole group. And so this week we're going to have an opportunity to review it and then apply our learning in a very specific individualistic manner. In the slides today, we're going to do a review, so I'll spend about 10 or 15 minutes talking over meiosis and mitosis again. You'll have an opportunity to practice identifying real live cells or images of real live cells, and then you're going to have an opportunity to choose which is which, so which is mitosis and which is meiosis. And then we're just going to get down to the straight point, and hopefully this will help you remember the difference between mitosis and meiosis. So without further ado, let's go ahead and begin. So what did we learn last week? So if you're a Taylor Swift fan like I am, then you should be familiar with this lyrics. We are never, ever, ever getting back together. And I saw this and obviously my biology nerdy self thought this was hilarious. And so think about what it's saying and why is this a joke? What's the meaning behind this, right? So taking a look at what we're learning and what it's saying, what do you think it is? Once you think about it, you can let me know in next week's small group. So as we move forward, there's a couple of vocabulary words that I'd really like to point out because there's a lot of stuff about DNA, chromosomes, what are the differences, right? So I want to spend a couple of minutes only talking about the vocabulary words that we're going to be using in this unit. And so we talk a lot about interphase, right? So interphase is that part of the cell cycle that is made up of three parts. That's the G1, which is the growth and formulating or going out and doing the normal functions. There's the synthesis, which is when they're going to be duplicating DNA, and G2 when it starts to get ready for it to go into mitosis. So that is what interphase is made up of, and it is what the cell life lives its majority of its life in. So that's interphase. We're going to go ahead and I'm going to talk about DNA and then genes, chromosome to chromatid. So first of all, DNA is the backbone. It's the genetic backbone. It has all the information and it contains genes. So if you take a look at our screen, it is this thing that is in the middle of the page, right? It is this guy. This entire thing is what we think about as the DNA. And this is what we consider the backbone of our genetic information. And so when we think of the DNA, we're really thinking of that as the place where it holds all of our genes. It's the place where we have our information, tells our cells what to do, what to create. And so that is what the basis of it all is, right? So let's skip over to genes. So we talk about DNA, we talk about genes, and what is the difference? Well, the difference is that the genes are encoded in the DNA. They're a segment of the DNA. So it's like this entire segment right here. I just really like these markers, so I'm just going to use them. This, for an example, would be a segment of the DNA that has a code that tells the cells what to do. And so when we talk about genes and DNA, really the important thing is for you to know that a segment or a part of the DNA is the gene. Genes are encoded, okay? So think about code, think about, how we use only a small number of letters to represent something larger. And the order of that is going to tell you what the code is, right? It's going to tell you those genes. So that's how genes and DNA are related. Genes are embedded or encoded in the DNA. So then what's the relationship between DNA and chromosomes? So remember that when you're thinking of chromosomes really only come up during mitosis and during meiosis. And think about it this way, right? If you think back to when you had your earbuds that had the string, you know, before we got all fancy with the AirPods, think about that string, and when you would put it in your backpack, if you didn't have it nice and tightly wound, it would just kind of string along, and if you wanted to move it from one backpack to another and you pulled it out, it would get caught on a bunch of other things, or even get knotted on itself, right? So the best way to move your earbuds are from, you know, winding them up, whether you did like one of those like fishnets and then you kind of wound it around your finger or whatever it was, but you did it in a way that would condense it so that it was easier for you to pick up and move. It would be easier for you to get out of your pocket, out of your purse, your backpack, whatever it is. But by creating it to be more condensed, you're saving space and it's easier to move around. Well, that's the same thing with DNA. While it's an interphase, right, the majority of its life, the DNA is just kind of out, it's unwound, it's doing its thing. But during mitosis, when it's really important for those chromosomes to be going to the right locations with as little mistakes as possible, then they get packaged around each other. They get wound around these proteins called histones, and that is what you see as chromosomes. So it's really just those long DNA strands that are getting ready for mitosis and meiosis, and they are wound tightly, and that's what we see as the X. Now, those are our chromosomes. Now remember, during the S phase and interphase was when it was DNA synthesis. All of that DNA was duplicating. So we have two of the same strands. That is the chromosome. That X that you see, as we normally think of as chromosomes, that X is when we have those duplicated DNA. Once you get to metaphase in both meiosis and mitosis, you start to see that they get lined up, and then in anaphase, they get separated. Those chromosomes get separated, and that's what we call chromatids. Remember, double, right? They're the same. They're the exact same thing in mitosis. Meiosis one in meiosis is different, right? So in mitosis, they're the exact same. And so you have two sister chromatids, two chromatids that are identical, that are connected. And then when they get pulled apart, right, one goes to the cell, the other goes to the cell, complete DNA, exact same thing. And then they get separated, and those individual strands of a chromosome are called chromatids. Right? So that's kind of the connection there. Moving on to the centromere. The centromere is the point in the middle of the chromosomes, the chromatids, where there are joined. So when we're talking about metaphase and anaphase, we're talking about those spindle fibers, that's coming next, that's going to connect at those centromeres to pull them apart. And then we have the spindle fibers, and I bring this up all the time just because it is one of my favorite things. Like, I do love talking about the spindle fibers because it's so cool. They're called microtubules. That's what the material is, and they kind of extend from the centromeres to each of the centromeres to the chromosomes to connect to those centromeres to then pull them back. It's kind of like this fishing pole situation where it gets cast, you get hooked, and then you reel it back. But there is nobody casting the line. It just kind of goes out and kind of spins outward, and it's really cool. So those are some important vocabulary words to know. And so as we go into me explaining, I'm putting myself right in the middle, where we talk about mitosis and meiosis. And so the big thing here is I want to point out this idea of PMAT. Now, PMAT are the specific phases that fall under mitosis and under meiosis. Interphase is not part of mitosis, but we talk about it because it gets it ready, right? And then cytokinesis is also not technically part of mitosis, but it gets a little tricky because it happens at the same time as telophase. So it's not technically part of mitosis, but it kind of gets intertwined because it happens at the same time. So when we're talking about mitosis, a couple of things that are really important to remember. Mitosis is what we have, it goes through all of our body cells. So every cell except for the sex cells, right? So that's the sperm in men, the eggs in female. So in every other body cell that we have, or somatic cells, we go through mitosis. And there are 4 main reasons why we do that. One, the main purpose is we wanted to produce identical cells. That's one of the biggest things. We call those daughter cells, identical daughter cells. Second thing is it's for when things are growing. So plants, when they grow, they're going through mitosis. When us as bodies are growing, we go through mitosis, right? The third thing is to repair any damaged cells. So for example, I talked a lot about my burn. It's in much better condition now. And my entire healing process was going through mitosis. It was replacing those damaged or repairing those damaged cells. And it's also used for number 3, it is used to replace dead skin cells. Okay, so that is, or to replace dead cells in general. Remember, each cell, this is part of your cell differentiation lesson, each cell has a specific role, and depending on the role of that cell is going to determine how the life of it. So some cells have a shorter lifespan than others, for example, skin cells. So we have mitosis to replace the dead cells, right? And so those are the major things. And so when we're going through mitosis, it's going to go through that PMAT process. It's pretty straightforward. So you're going to have an interphase, the cells are duplicating, and then when it gets to prophase, that's when the chromosomes are starting to be formed, that DNA is wrapping up and being condensed. The nuclear membrane starts to break down, and those spindle fibers start to form. So that's all what's happening in prophase. In metaphase, the chromosomes line up in a single file line in the middle of the cell. Got it? Metaphase, middle, right? So kind of thinking along those lines. The spindle fibers are connecting to those centromeres, right? And so they're getting ready in order to split apart, which they do in the next phase called anaphase. Anaphase is when the chromatids get separated and pulled away. Anaphase, away. And this is where that word chromatid happens, right? So the key thing here is that in mitosis, the chromosomes are lining up in a single file, and then the chromatids get pulled apart to the opposite ends of the cell, which is then when telophase occurs in cytokinesis. In telophase, you start to see the nuclear membrane starts to reform, the spindle fibers start to retract, and then cytokinesis is happening where in plant cells, they create a cell plate, so they start to build a wall up to divide it, and then in animal cells, those cells actually start to pinch off and they start to cleave. And the results are diploid cells. Remember, diploid cells are when they are all of the chromosomes your body needs. And so in humans, that's 46 chromosomes. But in meiosis, how it's different is this is just for when the purposes of reproducing. So that's why we talk about sperm in men and eggs in women. So similar to mitosis, meiosis is going to have a similar pattern, but quite different and unique in its own right. So in meiosis, the first thing is prophase 1. And prophase 1 is amazing. That's when you have the crossing over. So that's when, remember, over in interphase, you were having the DNA was duplicating. And we get these X's of double the genes, right? And then when you get these chromosomes in prophase 1, they start to mix and they start to cross over or swap their genetic information. You can kind of see in the picture here. So this is prophase 1. The nuclear membrane is breaking down. The spindle fibers are forming as well. Crossing over is really what's going to make it unique and is the cause and the leading factor for genetic variation. So that's how we have just such a variety when it comes to looking different from your siblings, looking different from your cousins, how we have different shapes of noses, lips, eyes. All of those things are all attributed to prophase 1 for crossing over. So then it goes into metaphase 1. And a couple things to point out here, if you can see kind of closely, that's when you start to see a little bit of those genes kind of swapping. You get a little bit of pink on the purple and a little bit of purple on the pink. Metaphase 1, you start to see that the chromosomes aren't lined up in a single file, but instead they're lined up coupled together. And that's a major difference because what is going to happen in anaphase 1 is that the entire chromosome is going to be pulled to the other side, not chromatid. And that's when we start to end up with haploid cells, or half the chromosomes, right? So in humans, that's 23 chromosomes. So remember that in anaphase 1, the chromosomes are pulled. It goes through telophase 1, so the cells are splitting, and then cytokinesis that, completely severs the cells. And then it's going to go through the process all over again, but this time in prophase 1, it doesn't go through crossing over. Now it starts to look more like mitosis. So it's going to go through prophase 2, which is when they condense, the nucleus starts to deform again. It's going to go through metaphase 2, where they are going to line up in a single file. They're going to go through anaphase 2, which is when the chromatids are separated, and you have, and then goes through telophase. Telophase 2, which is when you get them to separate and the nucleus starts to reform, the spindle fiber starts to go away, and then cytokinesis, which then you result in 4 haploid cells. And each one is genetically different from the other. And that's how we get meiosis. And that's why you can look different from your siblings, because you don't necessarily get the exact same genes, right? So that is a brief overview of meiosis and mitosis. Okay, but now you get to do the fun part. So on your version, on your copy, you are going to get a screen like this, and I want you to go ahead and practice putting the images of mitosis in order. So what is interphase, prophase, metaphase, anaphase, telophase, / cytokinesis? Okay, so you can pause this video, go ahead, try it on your own, and come back after a couple minutes to see if you got it right. Okay, so if you're coming back, you're going to notice here are the answers. Let me go ahead and move my face. But this is the order of mitosis. So we've got our interphase. You don't see the chromosomes are uncondensed. You can't really make it out. The nuclear envelope is there, right? You have some centrioles, centrosomes with centrioles. And you see the nucleolus, right? And then it goes into prophase. The chromosomes are condensing. The nuclear membrane is breaking down. Those spindle fibers are starting to form. Then you go into metaphase. They're lining up in the middle. The the spindle fibers are connecting to the centromeres. In anaphase, they get pulled away. And then in telophase, we start to see that they start to unwind again. The nuclear envelope starts to form. Cytokinesis is happening at the same time because it's starting to cleave. So if you got it correct, awesome job. And if you didn't, just take a look at what you did and think about it in like a logical step-by-step, right? You can't pull them apart before they are perfectly lined up, right? So taking a look at those steps and try to understand what logically makes sense instead of just memorizing it, right? So if you finished, great job. Let's see how you do when we're just talking about the characteristics, right? So now we're not going to be using the pictures, but instead we're going to be taking a look at these boxes. And I'd like for you to pause this video now and go ahead and put the boxes in the correct stage of mitosis of when this is happening. All right, good luck. Now that you've come back, here are the correct answers, right? So remember that we have the copies are being made, chromosomes are not visible, chromosomes are unwinding, prophase, the nuclear envelope breaks down, the chromosomes are condensing into X shapes, the spindle fibers start to form, In metaphase, those chromosomes are lining up at the equator. The spindle fibers are attaching to each chromosome. Anaphase, they're getting pulled apart. Telophase, the chromosomes start to unwind again. They divide into new cells. The chromosomes are nearly visible, or not clearly visible, and the nuclear envelope reforms. So if there were anything that you got wrong, take a look and reflect. Why did you think that? And does it make sense now that you've had a chance to take a look at what the answers are and thinking about what happens in each phase? If you got it correct, awesome job. You're doing great. Either way, we're learning, and that's the most important thing. Let's go on to the next one. So these are the 4 important things, right? The purposes of mitosis. And I've got these 4 lines, and I want you to take a look at the clues, right? So go ahead and type this into your slide. Pause right now so you can go ahead and do it, and then press play to see if you got the correct answers. Welcome back. So we have the correct answers. So remember that the purposes of mitosis is to grow, to repair damaged cells, to replace dead cells, and to have identical cells. So the plant was an example of growth. Don't forget that this is to all organisms, not just plants. The nose, the scar on the lion's nose was supposed to represent the damaged cells. Skin cells, because they're the shortest living cells, among one of the shortest living cells, and they replace it, and then the daughter cells to show that they're identical. All right, now into meiosis 1 and 2. I already have these filled out for you, but they're really just kind of reiterating what I went over at the start of the video. So remembering that meiosis specifically talks about sex cells, right? We call those sex cells gametes, and they look like eggs in women and sperm in men. And they have 23 chromosomes, right? Moms or dads. And so that's what we call half of them, right? Crossing over happens in meiosis when chromosomes mix and the genes are swapped during prophase 1. The end of meiosis 1 produces 2 cells with half the chromosomes. We call these half the chromosome cells haploid cells. And at the end of meiosis 2, we have 4 unique genetic cells. And then some information is we call human somatic cells have 46 chromosomes, which does not go through meiosis. Now go ahead, try one more time, pause the video, and now put meiosis in order. And this does encompass both meiosis 1 and meiosis 2. Good luck. If your order looks something like this, then you got it correct. If you didn't, take a look and see what is different about yours than it is mine, right? So we really want to be reflecting on our learning. It's okay if you don't get it right. I don't expect you to get it right the first time, because part of learning is making those mistakes and then recorrecting or rewiring your brain. And the more and more practice you have, the more and more you're going to remember and learn it. So no worries if you had a couple mistakes, but take a look and see where those mistakes are and think about why. Maybe you had the images reversed in your mind, right? So again, we want to take a look at the logistics, right? The logical order of things. You can't separate until you're already lined up in the middle. You can't line up in the middle until everything is nice and condensed, okay? When you do separate, you've got 2 cells that have full chromosomes, and then you got to do the process again. So these guys are in the middle, can't separate until you're in the middle, can't separate, right? So now you're separated. You can't divide until you're separated, and then your results are 4 haploid cells that are genetically unique. Good. Now let's try looking at real life cells. Okay, so these are images of cells in an actual slide. And so as you can tell, it is quite different than the drawings that we see in our textbook. And it is a little bit more challenging. So go ahead, we have a mixture of animal cells and plant cells, but take a look at these cells and try to put them in the phase that they are in. This one over here this is the only one that's interphase. It's already identified for you. So that means that the rest of them are going to follow PMAT. Couple of clues I'm going to give you. In this one, take a look. There are 2 dots that are dense. And I'm going to give you a hint. Those are 2 nuclei. So if those are 2 nucleus, what phase is going to allow you to have 2 nucleus? in the same cell. Try it out and click play when you're, pause it, try it out, and then click play again when you're ready for the answers. Here are the answers. So remember that hint that I just gave you? These have been the most difficult to classify because they don't exactly look like what we saw in the pictures. And even though it doesn't look like this one where you've got them cleaved, you do still have two dense dots, and those are signifying that those are two nuclei, meaning that they've already separated and are just waiting to go through cytokinesis. You've got anaphase because you can see that the chromosomes are splitting. These guys are in the middle. In prophase, you don't exactly see the chromosomes, but you can start to see how they're starting to split. The nucleus is starting to break down. Good job, guys. Now, second to last thing, I want you guys to identify these following statements under classifying them as meiosis or mitosis. There are nothing that's going to go in the middle. They're either one or the other. So go ahead and after everything we've learned today, which one is going to fall in each category? Pause. Press play when you're ready. All right, and these are the answers, right? So remember that the biggest thing about meiosis has to do with reproduction. Yes. Feel like that sounds weird. Yes, when you're trying to reproduce. There we go. Oh my gosh. When you're trying to reproduce, these are what's going to help you remember that, okay? So you want to, it creates germ cells or eggs and sperm or gametes, right? There's two successive self-divisions. Prophase one takes longer because we have that crossing over. Daughter cells are different. Haploid cells results in 4 daughter cells. Mitosis, the daughter cells are identical. There's only one division. Prophase is shorter. They're not crossing over. They just want to get that duplicated and split up. It results in diploid cells, creates body cells or somatic cells, right? Metaphase, and the chromosomes line up in the center. No recombination or crossing over occurs, and they result in 2 daughter cells. And so the best way to try to remember this is if you remember the point. And when you get down to it, mitosis is to create identical cells, and meiosis is for making babies. And so if you remember it that way, those are often the best ways that it's going to help you kind of think back to how each thing relates to one another. And that is your small group for the week. Thank you so much for joining me. I'm sorry I wasn't able to be there in person, but hopefully you had some success today and you've had opportunities to learn. Thank you so much. See you next week.